Hello everyone. So we the students of Central University Kerala pursuing a master's in Department of Plant Science. Today we are going to do a review on the research article. I am Karan, this is Dana and Kamal MJ here. So this article is taken from the Plant Journal. The title goes like this. Abscisic acid enables germination of Striga seeds and is released by them likely as a rhizophoric signal supporting host infestation. The authors are Mohammed Jamil, Yagis Lagos, Jianyu Wang and others. This was published in the year 2024. Let's discuss the introduction part. Abscisic acid, ABA, is a plant hormone known for its regulatory role in various growth and development processes. It also includes some of the stress responses. In the context of Striga hermantheca seeds, which are the root parasitic plants, ABA plays a crucial role in modulating seed germination, and the Striga seeds also exhibit dormancy to avoid germination under unfavorable conditions. But it can also respond to germination stimulants released by the host plants, and particularly Striga lactones, which is SLS, under suitable environmental conditions. This study aims to investigate how ABA influences the dormancy and germination responses of Striga seeds, shedding light on the early interactions between the parasite and its host plants. So overall, ABA serves as a rhizoporic signal, likely facilitating communication between Striga seeds and host plants, supporting the host infestation. And now let's discuss about the materials and methodology. The study utilized key materials such as floridone, RAC GR24, which is a synthetic SN log, ABA, and Arabidopsis ABA reporter line to investigate the role of ABA in Striga seed germination. Experimental techniques includes seed germination assays to assess the effects of different treatments, ABA quantification methods to measure hormone levels, and gene expression analysis to understand the molecular mechanisms underlying ABA responses in Striga seeds. The researchers employed a range of tools and methodologies to study how ABA influences the germination process of Striga seeds.
So now let's discuss about the experimental procedures. The first one is hormonal profiling. This procedure involved quantifying plant hormones in Striga seeds using a specialized protocol. Ground seeds were treated with the internal standards and subjected to sonification and centrifugation. The resulting supernatant was processed, dried, redissolved, and analyzed using LC-MS techniques. By following this method, researchers could accurately measure hormone levels in the seeds. The second one is plant hormone analysis. The quantification of plant hormones in seed exudate was carried out using LC-MS with the MRM mode. The specific chromatographic conditions and mass spectrometry settings were optimized for accurate hormones detection. The analysis focused on ABA levels in the exudates and provided insights into hormone dynamics during the seed conditioning. The third one is gas sustaining biosynthesis and imaging. Arabidopsis seedlings were exposed to different treatment, including water, ABA, and Striga seed exudate. Following treatments, seedlings underwent gas staining assays to visualize gene expression patterns. The staining process involved specific buffer incubation, de-staining, and imaging to assess the effects of the treatment on gene expression. Moving on to the next experimental procedure, that is gene expression analysis. The stigma seeds were subjected to various treatments, and total RNA was extracted for gene expression analysis. The extracted RNA was used for cDNA synthesis and subsequent QRT-PCR analysis. The gene expression levels of specific targets were quantified and compared between different treatment groups. Statistical analyses were performed to determine significant differences in gene expression levels. The fifth one is modulating Striga seed conditioning and germination process. The ABA was identified as a key regulation of Striga seeds condition and germination. The study highlights the role of ABA breaking seed dormancy during conditioning and its subsequent release upon germination. The increased ABA biosynthesis and release were associated with a decreased ABA sensitivity, impacting seed germination and host investigation process. And the sixth one is role of abscisic acid as a rhizosporic signal. The ABA was shown to function as a rhizosporic signal in Striga communication. The released ABA facilitated the investigation process by promoting host root growth. The study emphasized the importance of ABA in early interaction between Striga seeds and host plants, providing a comprehensive advantages for germinated seeds.
The research led to the inference of four major findings. The first one, ABA inhibits seed germination and delays conditioning of Striga seeds. And the sensitivity to ABA decreases during Striga seed conditioning, And also, the strigolactone-induced seed germination triggers ABA biosynthesis and its release. And ABA released by germinating striga seeds delays conditioning of germination of the surrounding seeds. The given graphs represent the analysis of all the experiments conducted. The researchers investigated the role of ABA in striga seed dormancy and conditioning. They treated unconditioned striga seeds with fluoridine, that is an inhibitor of ABA activity, in the presence and absence of a strigolactone analog. Each chemical alone induced germination in 8 to 13% of the seeds, but when combined, they triggered germination by 70% of the seeds, suggesting that inhibiting ABA biosynthesis initiates germination and shortens conditioning. Striga seeds released ABA during germination, and transcript level of Striga CYP707A homolog, which catabolize ABA, were measured. Early in conditioning, Striga seeds release ABA unaffected by strigolactone application. However, in the late stage of conditioning, strigolactone application induced germination, leading to the accumulation and release of endogenous abscisic acid. Further analysis showed that ABA acts as a germination inhibitor, particularly in unconditioned striga seeds, with germination decreasing as ABA concentration increases. Seed conditioning for 8 days were less inhibited by ABA compared to those conditioned for 2 days. The given images explains how the researchers explored that ABA could be utilized to combat striga infestation. They investigated suicidal germination, a method involving the application of strigolactone analogs to the striga infested fields in the absence of a host, resulting in the death of emerging striga seedlings and soil decontamination. In suicidal germination tests, the authors induced germination of an unconditioned striga seed using a strigolactone analog, fluoridine, or a combination of both for 2 weeks, then grew rice as a host to assess striga emergence and host plant performance. Compared to the untreated pods, fluoridine treatment reduced striga emergence and resulted in healthier host plants. Treatment with fluoridine and the strigolactone analog produced similar outcomes. This indicates that abscisic acid inhibitors can act as suicidal germination agents by breaking seed dormancy even before conditioning. When combined with other hormones such as strigolactone, this treatment could be even more effective in combating striga infestation in the field. To assess the impact on surrounding striga seeds, the researchers positioned striga seeds in days 0 to 7 of the conditioning phase around a disc covered with germinated seeds or a disc soaked with abscisic acid as a control. Both discs inhibited the germination of surrounding conditioned seeds with the highest inhibition observed in seeds containing for a short duration. This mechanism likely provides a competitive advantage of germinated seeds over unconditioned or early conditioned seeds, maximizing the resources exploitation. Additionally, placing germinated striga seeds next to host roots promote root growth and increase the number of lateral roots growing towards the seeds. Thus, abscisic acid not only plays a role in endogenous in conditioning and germination, but also in exudates to manipulate the surrounding seeds and host root architecture. The increased number of host lateral root growing towards the abscisic acid released by germinated striga seeds might enhance the likelihood of striga attachment to the host root system. This image shows how the signaling works between the host and its parasite, and also about the lateral root growth.
This is an illustration and model showing on the role of AB in striga germination and host infestation. Conditioned striga seeds germinate perceiving host-released SLS, that is, strigolactones. Germination initiates the development of a hostoirium that enables the host infestation and then promotes AB in them. AB released into this ribosphere negatively impacts the conditioning and germination of the surrounding striga seeds, and this promotes the growth of host roots towards the growing parasite, providing a competitive advantage and supporting host infestation. With this, we come to the end of the session, and talking about the inference, in conclusion, the study underscores the essential role of ABA in regulating the dormancy and germination process of striga seeds. ABA serves as a key player in modulating seed responses to environment cues and host plant signals, and this influence parasite-host interactions. The findings emphasize on the significance of the ABA as a regulator of seed germination and impact of communication and infestation process between the striga seeds and the host plants. And if you have any questions, please do post in the comment section. Thank you. Don't forget to post your questions.
Thank you.